Therapeutic Insulin Production, created by Jeff Falta and Taylor Libas. The synthesis of insulin is necessary for the treatment of patients with diabetes. In the past, cow, pig, horse, and fish were used as sources of insulin because the protein is nearly identical to human insulin. Yet other substances in the isolation elicited immune responses in patients. Also, the use of animals is not sustainable, as the pancreas of the animal must be destroyed. DNA recombinant technology is now widely used to produce human insulin using bacteria such as E. coli as a vector. This diagram shows how recombinant DNA technology works. First, plasmids are removed from the target bacteria. Second, an enzyme such as eco-RI endonuclease is used to cleave the plasmid. Third, the insulin DNA is cleaved by the same enzyme used to cleave the plasmid. Fourth, the insulin DNA is integrated into the bacterial plasmid through use of the enzyme DNA ligase. Fifth, the complete plasmid with the insulin-producing gene is incorporated into the plasmid. These insulin-producing E. coli replicate to form additional E. coli bacteria and are able to produce human insulin that can be isolated and used for the treatment of diabetes. In order for the bacteria to grow and replicate, the chemostat conditions must be maintained and tightly regulated. These conditions include the temperature, pH, the media formulation, the oxygen content, dilution rate, and agitation speed. Some of the ingredients for the media are shown here per 1 liter of media. Note that the formulation of media must be tightly controlled. Insulin is produced in a bacterial chemostat at Sugar Regulators Incorporated. One day, a flow controller which controls the flow rate of the yeast extract stopped functioning correctly. The process engineer took samples every minute for 100 minutes to determine whether the concentration of the yeast met the design specifications. Concentration of the yeast must be in the range of 4.95 to 5.05 grams of yeast per liter of media. The sample data was noted to follow a normal distribution. Help the process engineer determine the probability of whether the media will meet the design specifications. 100 samples were taken from the chemostat over a 100-minute time span. The concentration of yeast ranged from 4.93 to 5.07 grams of yeast extract per liter of media. The frequency of the sample data are recorded in the table provided here. The mean value of this data is 5.00 and the standard deviation is 0.03. This graph plots the frequency of sample measurements versus the concentration of yeast extract. The sample data appears to follow a normal distribution. In order to determine the probability of whether the media will meet the design specifications, the following method is used. The first equation represents the probability density function. In order to determine if the concentration will fall within the specified range, the probability density function is integrated over the specified range, which results in equation 2. Since equation 2 may be difficult or impossible to solve, equation 3 is used because it utilizes the error function. Equations 4 through 7 carry out the mathematical calculations. Therefore, the process engineer concluded that design specifications will be met with 90.5% confidence while the damaged flow meter is repaired.